My name is Sam Norman Hegner, and I'm a postdoctoral associate in Nancy Canwisher and Josh McDermott's lab at MIT.
My lab is interested in the overall organization of the human brain. What are the parts of the brain, and how do they relate to parts of the mind? That's why I'm interested in this. I want to know who we are as thinkers, what the basic pieces are of the human mind. And I think we can see those by looking at the organization of the brain.
So at present, sort of surprisingly little is known about the basic organization and functional properties of auditory cortex, which is the part of the brain that responds to sounds and involved in processing music and speech and other complex environmental sounds.
So there had been many proposals for how human auditory cortex might be organized into regions and their association with function, but really very little consensus. So the goal of this study was to try to step back and take a very broad view of how human auditory cortex might be organized, and to not really be particularly reliant on any prior hypothesis about that organization.
We measured responses using an MRI in 10 different human subjects. So what that means is that we put someone in the scanner and we played them sounds while we measured responses with an MRI machine. So in practice, we measured responses to 165 different sounds, like for example, the sound of a toilet flushing, or the sound of someone shuffling deck of cards, or the sound of someone walking. These are just sort of the sounds that we hear in everyday life. And so the result of that experiment is that basically we get a matrix. And what that matrix tells us is it tells us for every point in the brain, how much of that point of the brain respond to each of the 165 sounds.
What emerged from the analysis was that there's a relatively small number of functional properties that seem to dictate the organization of auditory cortex. When we find that there's about 6 of these canonical components that seem to be able to account for the responses across auditory cortex in different proportions in different parts of the brain.
The first 4 components that emerged from this analysis were aspects of the neural response to sound that we sort of anticipated from other work. But 2 of the components that just popped out of this very broad screen were striking selectivity for speech sounds and striking selectivity for music. And that really blew our minds.
So this result was surprising to us for several reasons. One is that when I first came to grad school, I spent basically a year or 2 looking for brain regions that respond selectively to music, and basically failed to find them. So going into the study, I expected not to find clear selectivity for music. And the explanation for why we were able to see this clearly with this method as opposed to other methods, including methods that we had previously used, is that we think that the raw data of fMRI is spatially coarse, and that as a result you need to model the raw data, you need to model voxels as reflecting multiple things, in this case multiple neural populations. And that modeling procedure effectively allows you to resolve patterns of selectivity that are not able to be seen very clearly in the raw data.
So it's extremely interesting to find some evidence that in fact there does seem to be neural machinery that is specialized to some extent for music perception. Now what we still don't really know is like where that comes from. Is that something that humans are born with? Is that something that emerges over the course of development because we get lots of exposure to music and it's an important and interesting signal? There's a lot of really deep and open questions that I think really speak to fundamental issues about where music comes from, why we have it, and how if it evolved if it did. And so I think the ability to see this neural population potentially gives us a method that we can use to get some leverage on those questions.